So first of all, I would like to thank organizer and scientific committee of Mustafa Foundation for inviting me. And then I would like to thank Professor Azad Manesh for his very nice and informative talk. I'm Mehdi Totonchi. I'm in charge of diagnostic lab at Royan Institute as a NGO organization in Tehran. So I'm also running my own laboratory in the field of mainly clinical bioinformatics, NGS data analysis, and protein docking. So today I would like to do, I'm going to talk about the profiling of SARS-CoV-2 sequencing in the data from Iranian related COVID-19 patients. So as an outline, we will go to coronaviruses classification, the history of this virus. Then we will go to the phylogenetic tree. Then we talk about this global outbreak around the world as well as Iran. Then I will share our result regarding profiling of this SARS-CoV-2 in Iranian patient. So coronavirus are related to the family of coronaviridae. They contain a single strand RNA of 26 to 32 kilobases and have been identified in several avian and mammals hosts. So they are they genetically divided into four types: alpha, beta, gamma, and delta coronaviruses. Mainly, the virus which are infected mammals are contained in the beta coronavirus, including CO43, SARS, and MERS. As an history, you can see we encountered these viruses since 1966. We had two of these coronavirus around the world in 1966 to 1967, but in general, they didn't have any severe symptoms around the world. But in 2002, we encountered SARS-CoV, which belonged to the beta coronavirus. The receptor its receptor in human was ACE2, and its natural host was bat. Unfortunately, we had around 8,000 infections around the world, with nearly 700 deaths at that time. So during 2 decades, we encountered 2 large or huge pandemic against these viruses. One in 2002, and the other in 2020, which was about MERS coronavirus. The host was bat and camel, and as it recorded, more than 1,000 human infections and around 600 deaths was emerged in the 27 countries. So, but what about the novel coronavirus? From December 2019, the patient with typical clinical symptoms such as fever, dry cough, breathing difficulties, headache, and pneumonia was emerged in the Wuhan, China. According to the data which we have, most of these people had a historical relation with the seafood in Wuhan. So after some clinical and genetically test based on next generation sequencing and de novo sequencing, we saw that we have a novel coronavirus in China, so which was around 80% identity with the previous one, SARS-CoV. So this new infectious respiratory disease is called coronavirus disease 19 or COVID-19. So what about the sequence of this coronavirus? From 5' to 3' of this genome, as I said, this is a single-strand RNA. There are many ORF, ORF1AB, ORF1A, which has around 7 non-structural proteins, including leader protein, ORF1AB, 3C-like proteinase, and RNA-dependent RNA polymerase, helicase, 1 exonuclease, endoRNase, and 1 methyltransferase. So regarding the 3' terminus, we have 4 structural proteins such as spike, nucleoprotein, membrane protein, and envelope protein. How we know this its genome characterization? When we compare the genome of this coronavirus with previous one, we saw there are 79% identity with SARS-CoV and just 50% identity with MERS coronaviruses. So whenever we compare the complete genome of SARS-CoV-2 with the other viruses, the BAT-CoV RATG13 is most similar or have higher similarity with this novel coronavirus than pangolin CoV with 91%. So this was a comparison or alignment with in the level of nucleic acid. Whenever you align its protein with other SARS-CoV, you can see the genes such as ORF1B, S protein, ORF7A, and ORF10 in pangolin depict higher identity with SARS-CoV-2. So there are some difference between nucleic acid and protein level. So there are some controversial that what is the intermediate host, and or if there is any gene pool between these coronaviruses. So regarding the global outbreak of this novel virus or SARS-CoV-2, up to end of the May, around more than 6 million cases we had, and unfortunately around 370,000 people expired because of this infectious. So what about Iran? Iran, up to end of the May, Iran is in order of 12 with around 1,500,000 cases and around 77,700 in end of the May. So some of the initiatives, such as GI said, are in charge of gathering and promoting and fast sharing of these novel coronavirus sequences. So up to end of the May, they gathered more than 35,000 sequences from this novel virus, which around 26,000 of them are full genomic. I mean, they had more than 29,000 bases. So as a natural evolution, this coronavirus, such as other viruses, has some mutations. So up to now, we can divide this virus to 5 categories, which you can see here. So why this is important to know how this virus can affect or have a spontaneous variation or mutation in its genome. So because it would be very important to know it because of the diagnostic test as well as in treatment. For example, based on different guidelines, Professor Azad Manesh said, we had different set of primers and probe which can target to have diagnostic tests. So whenever the viruses have a spontaneous variation or mutation, so we have to be aware about this changes to have lower negative, false negative results in our laboratory tests. So this is very important. So as a diagnostic test, we have to know, and as a treatment also, we have to know because, for example, regarding SARS-CoV, previous SARS-CoV, the scientists would wanted to target S protein to have some vaccines. However, they didn't able to generate very good vaccine for that at that time. But we have to know about this genetic variation or natural mutation in this regard. So based on this introduction, one of my postdocs, Dr. Salehi, was in charge of gathering the all available or initial available sequences which are related to the Iranian patients. So we had 3 steps in this regard: data collection from different database, especially GI-SAID and NCBI. Then we perform multiple alignment in the level of nucleic acid as well as protein with different framework and with different platforms. And in the, we did also protein structure in case of ACE2 to see how it, in case of S protein, to see how it binds to ACE2 protein. So in GI-SAID, at that time, there were 24 sequences which was related to the Iran. But just one of them had full sequencing, which was generated in Human Genetic Research Center. So we use this as our initial data, and out of this 24 data, just one was full length, and other 23 sequences of SARS-2 with a length of between 87 to 600. Just 15 of this were associated with N gene, 8 associated with ORF1AB, 3 leader protein, 4 RNA polymerase, and 1 3' to 5' exonuclease. And the same data from the NCBI. But we went to the NextString database, which is an open source project to harness the scientific and public health potential and pathogen genome data. At that time, we found some data which was generated around the world, but the patient was related, not located, related to the Iranian patient. It means the source of virus should be transferred from one of Iranian person. So we gathered this 19 data. We did multiple nucleic sequencing alignment, and we categorized into different levels which gene, which nucleotide affect or has more spontaneous variations. So in the level of protein also we did the same multiple alignment, which we translate the sequence in 6 different frames. So as a result, here you can see we had different nucleotide mutation here in different genes. We had so some of them had non-synonymous changes. So you can see here the different amino acid which was caused by this genetic variation. So here you can see the number of genetic variation in each location. For example, out of 20 data from Iranian related data, you can see here, for example, in ORF1A, 18 genetic variation caused. So we also compared the entropy of this data. The entropy values quantified the uncertainty and variability of amino acid mutation for each position in protein structure. So interestingly, there was some genetic mutation in S protein, which is the spike protein, which can bind to the ACE2 receptor in human cells. So there are some mutation, but none of them are related to the binding site. I mean, we conclude it should be there is no any effect to the protein-protein interaction in this case. So as a conclusion, we analyzed 20 full genome sequencing of SARS-CoV-2 from the Iranian-related COVID-19 patient that we gathered from different data set. So in the nucleotide mutation part, we saw there are common nucleotide mutation, especially in this location. We have to be aware that there are several mutations in N gene. So regarding the diagnostic test, we have to consider this. In the protein level, also we saw there are some amino acid changes in this mutation. So, however, we saw there are around 40 mutations and around 24 amino acid changes in this 20 database, but still we need more full genomic sequencing to have more information and accurate data to interpret for diagnostic part as well as treatment part. So we have submitted this result to Cell Journal as a short communication. It will be pop out in a few days. So finally, I would like to thank my colleagues in the diagnostic laboratory at Royan Institute for all their effort during this situation. I would be happy to answer any question you have. Do we have mutation in virus as an severity? So as I told you, we just analyzed 20 full genome sequencing which was related to Iranian patients. So based on that data, we found around 42 mutation in nucleic acid level and around 24 non-synonymous variation in protein level. So still there is up to there are just few mutation in S genes, but no in the other binding site of primer probe. So it means it does not affect to the accuracy or sensitivity of diagnostic test. And regarding the affinity of affinity or binding of S protein to the ACE2, however, there are some mutation in S protein, but it seems does not affect to this affinity. What caused the binding site mutations from bad cove so that is infect humans? What caused the binding site mutations from bad? What caused the binding site mutation from bad cove so that is infect humans? If I understood, you are looking for which part of this SARS-CoV-2 should have mutation to affect the human. This virus will bind to the ACE2 receptor. So if we have some mutation in binding site of this protein, so it should be somehow good, somehow bad, because it depends on how it will affect to binding affinity. So if I understood your question, this is so does SARS-CoV-2 have same binding site as bad cove? So in the level of can you show my slides? Here you can see. It's okay. It's okay. In case of ORF1b, S protein, ORF7a, ORF10. So the higher identity is between SARS-CoV-2 and pangolin. So, but in case of the rest of the genes, there are some. For example, in case of nucleic acid, you can see here there are more similarity with previous SARS. So based on this results, there are different gene pool in this novel or SARS-CoV-2. In case of ORF1b, S protein, ORF7a, and ORF10, the higher identities between pangolin coronavirus and SARS-CoV-2. But in case of the rest of genes, there are higher similarity between previous SARS and SARS-CoV-2. This one. Some countries like France have gene banks, so they can evaluate previous infection before November 2019. Excuse me. So, is there any chance for R to detect such a things in Iran? I'm not sure. Maybe Professor Azad Manesh can help me to answer this question, but I'm not sure if there is any good bank. But I guess we have to have this bank. So maybe Professor Azad Manesh, if you can answer. So here is.
We did this analysis. In fact, we checked something like a few hundred samples which were collected from influenza suspect cases during December and even October 2019, and we didn't find any positive sample before. I think the earliest one was something like the maybe 10th of February, I think. Not before that. So we have tested. We did not check the gene banks. We did study the sample, the clinical samples during the past 2 or 3 months, and they were negative. So there's no evidence for that.
Thank you, Professor. Thank you so much for the organizer, scientific committee, and all people to organize this nice events. Bye-bye.